Hello, I'm Mayumi from Kitasato University. So thank you for the opportunity to present our work about the RDF resources for biomedical research. So in this presentation, I would like to introduce our the concept of our project to integrate biomedical databases. So, as many presentations already mentioned, the integration of life science data is widely recognized as essential for drug discovery and medical research. However, it was also mentioned in yesterday's talk, the reality is many researchers, especially bioinformaticians, often integrate data manually with hand-copied in dataset. So one of the major challenges in integrating life science databases came from the multidimensionality and multi-layered property of data. So it is well recognized, especially in this community, that graph-based data representation, particular semantic web technology, RDF, is highly suitable for handling such complex data structure. And as you know, various databases are available in RDF format. This slide was kindly provided by Professor Katayama. And there are many well-known databases such as PDB and Uniprot, and PubChem, and also others. The Japanese flag indicates RDF data provided by DBCLS. On the other hand, biomedical databases such as those relevant to genomic medicine are often provided in the format specific to each database. And RDF data of those databases are not provided by original developers. So in 2017, we launched a project named Medo2 RDF in collaboration with DBCLS and Kyoto University and now Kitasato University. In this project, this project aims to propose data model to integrate key biomedical databases with other databases, and also we provide software tools to convert these databases into RDF format. So here is a list of databases we have targeted for now. So they include ClinVar, such as ClinVar, which provide information about disease-associated genomic variants. And here is also databases related to cancer and also predicted score of genomic variant effect. In working with these databases, we have also organized the necessary ontologies. The Medo2 RDF ontologies define the minimum required element to represent biomedical data in RDF. The GVO, genome variation ontology, offers a representation for various genomic variation identified in large-scale analysis, such as Nomad SV. For unified representation of human and mouse genome, the chromosome ontology HCO and MCO were developed. As you know, the length and the sequence ID in major databases of human and mouse chromosome are different depending on the built version, such as GRCH37 and 38 for human. So these ontologies provide simple and stable URIs for each reference genome across different databases. So we have developed the program for converting each data into our proposed data model with the ontology in the previous slide, and we shared them on GitHub, this repository. So development of this program has been carried out by volunteers from academic and also collaborative work with companies. A variety of programming languages have been used, including Python and Ruby, Rust, and others, depending on the expertise of the contributor. So while we have developed ontologies and software to handle major biomedical databases, we have encountered several challenges when trying to apply them in practice. So when using RDF data, the first step is to understand its data model. However, while the original developer, it means combatant developers, may grasp the model, it often takes many times for others to understand the data model. Additionally, since the development of software were carried out independently, so now we faced maintenance difficulties. As you know, biomedical data is frequently updated, requiring continuous maintenance. However, sometimes we cannot reach the original developer, combatant developers, leaving the software outdated. So to address this issue, if we can simply define the data model and adapt unified conversion framework, we can improve RDF data usability and simply those maintenance. So at DBCLS, a framework called RDF-config has been developed. The detail of RDF-config will be presented in the upcoming keynote by Professor Katayama, so I will just introduce the framework briefly. With RDF-config, data models, data models and conversion rules are defined in YAML format. For RDF data, by using the data model definition file, it enables us to automatically generate SPARQL query and schema diagram. Additionally, while currently limited to CSV and TSV format, this framework allows conversion original data into Turtle and JSON LD. In the Methods RDF project, we are currently adapting 4 databases listed here to utilize this framework for RDF data conversion. So we are preparing to make the RDF data generated by this project available for everyone to use. Also, this is limited to databases with permissionable licenses. We plan to publish SPARQL endpoint through RDF Portal, maybe from this URL. So the detail of RDF Portal and the latest update of RDF Portal are already presented by Dr. Shuichi Kawashima in yesterday's and today's poster presentation. So I'd like to skip the detail here. So I have introduced Methods RDF project for integrating biomedical databases. The aim of this project is to integrate data to support and promote biomedical research. In many research, integrated data is mainly used for machine learning approach and also data science. So let me introduce some of the ongoing works on how the data obtained in this project will be used. So the first project I'd like to introduce is related to variant interpretation. So a little bit busy slide, sorry. And Fugaku is another name of Mount Fuji. So I think many of you have seen this illustration before. So it is a part of a collection of illustration of Mount Fuji called 36 Views of Fugaku. The Fugaku is also the name of Japan's most powerful supercomputer, jointly developed by Riken and Fujitsu in Japan. In this project, using supercomputer Fugaku, we estimate the impact of genomic variation on protein function and drug interaction by molecular dynamic simulation, MD simulation. So genomic variants from source such as like CRISPR are mapped onto protein 3D structures, and molecular dynamic simulation are performed. So far, we have calculated over 20,000 variants, and currently we are constructing a public database to share calculation result. So previously, mapping variant to structure, this process were done by integrating various datasets using some script by manually. But to maintain timely and efficiency, we have changed this process to utilizing RDF framework and methods RDF dataset. Additionally, beyond the integrate genome data and protein structure data, we are also working on integrating another biological data, as shown here, using RDF. Furthermore, we are developing some program to extract table data from integrated RDF dataset, making it suitable as input for various data science analysis, such as prediction model using machine learning. And second project is related to drug discovery. Unfortunately, detailed information is currently available only in Japanese. The previous project is also only Japanese. Sorry about that. This project is a collaboration with wet lab researchers. We collect multimodal bioactivity data for various compounds, especially for natural product. Then collected data apply machine learning to construct machine learning and AI to construct latent space for discovering new drug seed. Then experimental researchers synthesize the proposed compound by AI, then repeat the experimental process, this validation process. In this project, my team is responsible for organizing the data generated by wet laboratories, integrating them with public databases, and provide structured data for machine learning group. Currently, we are working on integrating original data with public databases, such as PubChem and Chembu, utilizing methods RDF ontology and dataset. In both of the project I introduced, we extract necessary items from RDF data and integrate them into table data, then use it for machine learning and simulation and data science, data analysis. Currently, each project developed its own separate programs, but we are now working with DBCLS to develop a generalized framework that can be applied this flow more broadly. So selecting item and extracting data are done by SPARQL. So I think Professor Katayama also will present about this technology in his keynote, so please let me skip the details. So here is a conclusion. We launched Methods RDF project to integrate biomedical databases, utilizing some technologies developed by DBCLS to improve usability of the RDF data and to keep update integrated databases. And now we are trying to develop a generalized framework to apply converted RDF data to biomedical research. So here is acknowledgement. Thank you for your kind attention.